Hi, my name is Naila Littell, and today I'm going to be telling you about PCR and gel electrophoresis. These tools are very widely used for lab research, clinical research, paternity testing, and even recently in historical DNA fingerprinting. Our question today pertains to the bones found under the Friars Church in Leicester, England. We're wondering, are these indeed the bones of King Richard III, as it has been recently speculated?
In summer 2012, a body was found buried beneath a parking lot in Leicester, England, and it became widely speculated that the body was that of King Richard III. King Richard III died in battle in 1486, and while the body's skull was battered in a way that seemed to coincide with his manner of death, having been bludgeoned, scientists still had to be sure. The scientists at University of Leicester extracted mitochondrial DNA from the bones and went on to search for known descendants of the late king.
The king's sister had many children, and her last remaining descendants were discovered in Canada, and lucky for the researchers, they agreed to be subjected to DNA testing. The DNA of both subjects was submitted to PCR, or polymerase chain reaction, which uses thermal cycling to make enough DNA to get a clear picture of genotypes. The PCR heats the DNA to denature it, or separate the strands, then cools and allows primers and dNTPs to attach to the strands, doubling them in size. This process repeats 30 to 40 times, allowing the DNA to multiply exponentially. When there is enough DNA, a gel electrophoresis can be run, allowing researchers to see specific genotypes in the subject. The researchers likely ran these tests as well as others to get the DNA results of the two subjects, and then compared them side by side. When the DNA sequences of the body and the Canadian man were compared side by side, it was found that their sequences were nearly identical.
This man is without a doubt the great-great-nephew of the late King Richard, and the body can be given a proper burial.
Since their introduction into common use in the 1990s, PCR and gel electrophoresis have proliferated into many applications, such as the DNA fingerprinting that we have shown today, in the case of the body discovered under the Friars Church.